Hello and welcome to the DN Talk Show on this very popular YouTube channel, the DN News. The DN Talk Show is an exclusive talk with academicians, researchers, scientists, administrators, public and political figures, social activists, leaders with name and fame who are considered experts in various fields concerning issues pertaining to larger public of the nation. Coronavirus pandemic is not yet over, and India fights tooth and nail to eradicate COVID-19 disease from its roots. India, in its move forward, has come to the stage of producing vaccine to protect the people of this country from COVID-19. And we notice lot of major changes in the health sector with respect to developed infrastructure, facilities, hospitals, availability of drugs, ease of treatment to COVID affected persons. Today we have here with us Dr. Ravikiran Yedidi, who is a founder, principal scientist, and a lead instructor at the Center for Advanced Applied Biological Sciences and Entrepreneurship, Rajahmundry, and is a PhD in biochemistry from Wayne State University, Detroit, USA, and a person competent to speak on the true facts of the effects of variants and mutants of every moment germinating the fastly spreading coronavirus. Dr. Ravikiran Yedidi has deep research on coronavirus vaccine and is here to enlighten us on the dilemma, plight, and confused state of mind of the people in general with respect to side effects and after effects of the coronavirus vaccine. Dr. Ravikiran, we welcome you to our special program, the DN Talk Show. We are grateful to you for having spared your precious moments with us to create awareness amongst the people and to remove the confusion. I hope the program would be a meaningful one. Thank you very much. Thank you very much, Dr. Ravikiran. You are here on our show, the DN Talk Show. We will now discuss about coronavirus in general and also the vaccine that has come in India, that has been produced in India. There are a lot of confusions in the mind of the people. So we will discuss about that. First, we would like to know what is a virus, just a general outline, if you can explain us what is a virus and how is coronavirus different from those viruses which were already existing in the world. Can you please let us know about virus and this specifically the coronavirus? How is it different from the other viruses that it's not going despite having vaccines, despite having so useful treatment systems here in the country, but we are unable to fight with it? How does it make it different from other viruses?
Okay, good evening. Thanks for having me on the show, and it's actually a pleasure. And so basically a virus is a non-living organism, and the reason why I call it as an organism is because it has genetic material and all the proteins, lipids, and all those things that we normally have. So these viruses, they actually cannot live by themselves. They need a host. So it could be human being, it could be animals, it could be plants. So there are different kinds of viruses. So some of them, the different kinds of viruses are basically classified based on their genetic material. Some have DNA as their genetic material, just like the humans. Our genetic material is made of DNA. The genes are nothing but pieces of DNA. And some viruses are made up of genetic material, which is RNA. And so sometimes it is a single-stranded positive RNA, negative RNA, so that goes into depth, like molecular biology depth. But in general, layman language, so a virus is an organism that is invisible, so we cannot see it with an ordinary microscope. We need special microscopes, electron microscopes. It is so tiny, but it is something that needs a host in order for it to replicate and multiply and become more viral particles. And so the second question that you asked me about is how is coronavirus different from the other viruses? So first off, it's an RNA virus, so the genetic material in coronavirus is made of RNA. So the unfortunate thing is, if the viral genetic material is made of DNA, then we actually buy some time because that viral DNA has to come into the human cells, and then that DNA has to get converted into RNA, and then the virus actually gets replicating, and then it starts dividing to destroy the human cell. But in the case of coronavirus, it's an RNA virus, so it doesn't have that extra step where it has to convert its genetic material because the genetic material is RNA itself. So it is ready to invade and replicate and divide. And one of the most interesting things about coronavirus is the virus specifically targets the receptor protein that is present on the surface of human cells. And it's called the ACE receptor. And unfortunately, this ACE receptor is very critical for a lot of different things in human beings. For example, your blood pressure is maintained by the angiotensin. So ACE1, ACE2. So the ACE2 receptor actually becomes a very critical protein that we humans need in order to maintain our blood pressure carefully and normally, and a bunch of other things. So for example, to keep the inflammation under control. So inflammation is nothing but the reaction of the immune system when it sees a foreign piece of protein or a foreign organism such as bacteria or virus or fungus or something like that. So the best example that I can give you is when you have allergies. So for example, some people have allergies against certain plants and plant products. So when they come in contact with these plants or the plant extracts or plant products, they feel itchy and they start scratching, and within 5 minutes you can see like a reddish tinge on their skin. So this is the immune system fighting back that antigen or whatever the protein that came out of the plant or the extract. So this is inflammation. You can I guess the simplest way to explain inflammation is that redness. So imagine happening inside, the same thing happening inside in your organs or in your bloodstream or lungs or heart. So this inflammation can be increased or can be controlled by the ACE2 receptor. And unfortunately, that is the receptor that the coronavirus has chosen to actually enter into the human cells, which is really interesting part of this whole coronavirus infection. So this is something that my lab is still working on because I don't want to plant any unwanted thoughts into the minds of the audience, but there's always conspiracy theories out there, and some people say that this is genetically engineered virus by some country. I don't want to name any countries, because I don't have any evidence. So the fact that this virus specifically targets the ACE2 receptor, it kind of puts me in a dilemma that could it be an engineered virus? Maybe. But like I said, I don't have any evidence to support that fact. So coronavirus is just a virus. It's a large family of viruses. They infect us throughout the season, throughout the year. And mostly they cause simple fever or the cold, cough symptoms. But this SARS coronavirus 2, this is a special virus that is deeply involved in increasing the immune responses by specifically targeting the ACE2 receptor. And it also comes to the immune system. Like people who are healthy with a healthy immune system, they should be able to fight it off very easily. So it's just a virus like any other viruses. And in fact, if I compare coronavirus with HIV, HIV is more problematic. There are so many headaches with HIV, and it's a permanent infection. Once you get an HIV infection, it is permanent. But coronavirus, it doesn't stay in your body. Once it is cleared by your immune system, it is cleared by your immune system, unless there is a new strain with a mutation that comes in. But then again, the human system, the human immune system is designed. It is an intelligent design. We should be proud of our immune systems. And also, actually, I use this as an example when I talk to my students. If you don't like somebody, then don't look at their face. Think about their immune system. Their immune system is as good as yours. So there's no difference between you and them. So please don't hate people. The immune system, it is one of the fantastic, most complicated, intelligent design that the human beings have. And unfortunately, the immune system is not something that could clear off the viruses or infections in a matter of seconds or minutes. Because it takes time, because it has to catch the virus, it has to analyze the virus, it has to understand what the viruses look like, and then it has to produce the antibodies that would completely neutralize these viruses. So this process actually takes time. In healthy people, that process may be, like 3 to 4 days, or maybe within a week. But as you grow old, or if you have other complications such as diabetes, which is one of the huge problems in Indian citizens, and also the blood pressure fluctuations, and obesity. Of course, obesity is one of the issues that is actually becoming more prominent these days in India. So these complications may actually play a role in reducing your immune system, which means the immune system may take longer. Instead of taking 3 to 4 days, it might take a couple of weeks or maybe even a month to actually look at the virus and understand the structure of the virus and then generate the antibodies. But once your immune system does that, then you're fully protected, because that virus cannot reinfect you anymore, because you're permanently protected from that virus. Unless, like I said, if there is a mutation in that virus that changes its shape, then your body gets confused, because even though you have antibodies against a certain virus, for example, coronavirus, if there is a mutation that actually changes the shape of the virus, so for example, people are talking about the spike protein. I'm sure most of you have actually looked at how the coronavirus looks like. It looks like a spherical ball. On top of it, it has all those spikes. So the spike protein is the one that binds to the ACE2 receptor, and that's what we target. So if there are mutations in that spike protein, even though if your immune system had made antibodies already, it would get confused, because now there's a new strain with a mutation. That mutation changes the shape of the spike protein. So that's why.
Well, that's exactly what I wanted to ask you about mutation and about the new strain that we are now. The thing the people are talking about, the new strain, the second wave, and the double mutation. What is this double mutation, and what is the new strain? And today there is a news that about 7 new variants have been identified near Varanasi in Uttar Pradesh. What is all this? Are these different from the one that we have witnessed in the last 2020, or it's the same thing with some additional features? What is it, and how does it make a new strain? How does it come out with a new strain? What is it, new strain?
So scientifically, I can explain it in one way. Before I go into the scientific explanation, just like normal talk, my question to you is: do you know why Dr. Jamil had resigned from the sequencing project that was actually doing the genome sequencing for the SARS coronavirus in India? I was actually shocked to see Shahid Jamil, sir. He stepped down from the project. The project was exclusively focused on analyzing the DNA sequences of all these viral strains to understand how the viral mutations are happening. So, like I said, I don't want to plant any bad ideas into the minds of people. But then, is there a reason why Dr. Shahid Jamil stepped down as the in charge of the project? So this takes me back to what I've said earlier. The main question that I am still not clear as of today is this a naturally evolving virus, or is it genetically engineered virus? I mean, naturally evolving viruses actually take a long time. And the reason why I say that is because in the case of HIV, that virus, every time when it replicates, it makes mistakes and it makes plenty of mistakes. So one replication cycle in HIV will produce at least 10 to 12 mutations just for one round. And that one round could be several times in a day, which means one HIV particle can generate hundreds and thousands of mutations in one day. That's because the replication process of HIV has errors, and the virus doesn't care about it. But when it comes to the SARS coronavirus 2, what really throws me off is that this virus is well equipped. I mean, it comes with its own replication protein. It's called the RNA-dependent RNA polymerase. So the RNA-dependent RNA polymerase, it's basically an enzyme that would replicate the viral RNA so that it can make more viral RNAs. And those RNAs will pack into new viral particles, and eventually they destroy the cell and come out into the blood. Going back to the RNA-dependent RNA polymerase, I have looked at the three-dimensional structure at the atomic level, not at the molecular level. I looked at the structure at the atomic level, and I'm convinced with all the scientific evidence that we have today across the globe, all the way from the Americas to the Asia. So it tells me that the RNA-dependent RNA polymerase, that's an enzyme that replicates the SARS coronavirus 2 exactly the same sequence from the previous sequence. And it not only replicates the same sequence, but it also has an additional function where it goes back and checks if it made any mistakes. So, for example, if you have a sequence, CCGG, and during the replication, if this enzyme made a mistake, instead of CCGG, if it replicated ACGG, so there is a mutation from C to A, right? So, but this enzyme is so good that it would actually go back and it would cut out that A, and then it would replace it with C. So it not only replicates the viral sequence exactly the way how the original virus looks like, but also has the capability of going back and checking the sequence and making sure that it is an exact sequence. And in comparison to HIV doesn't do that. It just replicates. It doesn't check its own sequence. It has mutations. So like I said, in one day, one HIV particle can generate millions of particles with hundreds and thousands of mutations. But when it comes to the SARS coronavirus 2, scientifically, from my understanding, the analysis that I have done in my lab, the literature that I have studied from the papers that were published by the scientists across the globe, it convinces me that it is not easy to get mutations in SARS coronavirus 2, especially because that virus has an enzyme. Enzyme is basically a protein. So that virus brings its own protein that not only replicates its genome, the RNA genome, genuinely without making mistakes, but also if it makes mistakes, it goes back and it would actually correct those mistakes. So this is what puzzles me a lot as a scientist when I look at this. People are saying that they found 7 strains, they found 20 strains, or there's 1 mutation in Malaysia, or there's 2 mutations in Italy. It's not that easy for the virus to actually get these mutations, only because it brings its own protein along with it that makes sure that the viral replication happens genuinely without any mistakes. And also, if it makes any mistakes, it will correct itself. With that being said, the mutations, when the virus is replicating, the mutations can happen anywhere in its genome. So genome is basically the total genetic material that the virus is bringing along with it, that is packed inside the virus. So when the virus makes a mistake, as in if there's a mutation that gets incorporated into the genome, and it will not affect the infectivity of the virus, or it's not going to be a problem for the human beings as long as that mutation is not in the spike protein. The reason is when the coronavirus enters the human body, mostly through inhalation of infected air through the droplets, the viral particles enter the bloodstream through the epithelial cells in the lung, the alveoli. And so, the only protein that is actually exposed on the outer surface of the viral particle is the spike protein. And of course, there are some other proteins, but the major protein that is exposed to the immune system is the spike protein. So let's say, for example, if the coronavirus decided to make mutations, or if the coronavirus decided to, evolve and change its shape and structure, even then, it is very hard because the mutation has to be present in the spike protein. If there are mutations inside the other proteins of the virus, that's not going to affect anything. So, the spike protein is the only thing that is very critical in this virus. And like I said, if there is a mutation, that mutation has to be in the spike protein. And that's not enough. Just because you have a mutation doesn't mean that's going to change everything. No, oftentimes one mutation is not enough. I mean, yes, in rare cases, there might be one mutation that could be the game changer. But usually when you have one mutation, that may or may not actually change the virus and or turn it into a super virus or anything like that. Okay, so there has to be multiple mutations. And you have to keep in mind if the virus is making multiple mistakes during the replication, in other words, it is including or incorporating multiple mutations during replication, then you have to understand that the viral proteins are going to kill the virus itself, because more mutations in the viral proteins, the less functional those proteins are, right? So let's imagine you have a four-wheeler. It could be a car, it could be a bus or something like that. When you have the four tires properly installed, you can drive as fast as you can. But let's say if I puncture one of your tires, I don't think you can move, right? So that's a problem. Maybe one tire you may pull the vehicle to a certain distance. But what if I puncture three tires? Then it's completely impossible for your four-wheeler vehicle to actually move with the one tire. So similarly, the more mutations that the virus is going to change or incorporate into its genome, the less functional the virus will become, which means it may not be able to replicate faster or as good as the regular normal ones, or it may not be able to infect the human beings as potently as the original strain does, or it could be just meaningless. It could be just circulating in the blood, but it's harmless. So there's so many things that could impair the virus itself. If the virus decides to make mutations, those mutations are actually bad for the virus itself. So that virus cannot survive. So that's one thing. Another thing is,
So you mean to say that this mutation, which is with this virus is mutating so many times, this is going to be beneficial for the humans, that we are suffering so much because of this virus? After a certain number of mutations, you think the virus will be inactive or maybe not be harming the humans, and it will be beneficial for the humans?
right? So not all the mutations are beneficial for the humans. So it's a 50-50 chance. So if it's more like a flip of a coin. You can have it's a 50% chance. So let's say if the virus accumulates a certain mutation. Now that certain mutation could be beneficial for the virus, which means it's bad news for humans. Or there's a 50% chance that it is beneficial to the humans, then it becomes bad news for the virus. So it's a 50-50 chance. So that's why I mentioned earlier, so the mutation, it has to be in the spike protein. Only if the mutation is in the spike protein, then it is bad news for the humans, because the spike protein may change its shape because of the mutation. And also, not all mutations change the shape of the spike protein. There has to be a critical, very susceptible area in that spike protein. If that area gets mutated, then the overall three-dimensional structure of that spike protein changes. And if that happens, then yes, it's bad news for humans because our immune system knows how to handle the original strain. But now, if you have a strain that has a mutation with a changed structure of spike protein, then the immune system has to reanalyze the mutant and make new antibodies against this mutant. So it takes another week or 2 weeks again. So you have to go through the regular infection, cold, cough, diarrhea, whatever the symptoms are. So you have to go through the entire cycle.
A connected question to this, as you said that the old virus strain, which was with the immune system understood, and now after the mutation, the immune system doesn't understand that. And my question is connected to this particular thing, that when the coronavirus vaccine has been produced in the country, or maybe across the world, in many places they have produced. Now, this was produced for the old strain. My question is very clear and very straight to you, that will the same vaccine work for the new strain after double mutation that has infected so many people and people are suffering so much because of that? Will the same vaccine work in the same manner with the new strain also? And if not, then what is the alternative?
So the answer to your question is yes. The new strain should be handled by the vaccine. So the vaccine should be able to handle the new strain. Uh, the reason being, so let's say we know that the vaccine design started probably sometime last year, maybe in the month of July or August, I'm sure. I remember this because I think the Indian government wanted to start the clinical trials in July and release the vaccine in August, which was a big shock to me. But then again, that's not related to our discussion today. But anyways, the when you're using the old fashioned, so let me get back to the basics of vaccine first of all. There are different ways to make a vaccine. So the oldest and traditional way is you take the organism, whether it is a virus or a bacteria or whatever it is. You take that whole organism and then you inactivate that organism. Some people do it by heating it to a certain temperature so that the virus or bacteria is inactivated permanently so that it cannot replicate or infect. But the structure of the virus, the three-dimensional structure of the virus or the three-dimensional structure of the bacteria is still intact. It's not going to change its shape significantly. Okay, so I mean, heating may change the structures a little bit, but not significantly. So this is a traditional way of making vaccine. And the modern ways is what we do is we take that particular protein that is exposed. So, for example, in the case of coronavirus, the only protein that is significantly playing a role in the infection and also the primary target for any of the immune system or for the vaccine is the spike protein. So what the scientists are doing is, just take that spike protein instead of taking the whole virus. You can just take that spike protein and inject it into the humans so that the immune system will think that there is a viral attack so that it can make antibodies. And then, the latest is to actually use DNA vaccines and RNA vaccines coming from the United States of America, from the Moderna company. They have successfully evaluated in collaboration with the National Institutes of Health in Washington, D.C., that, they've successfully raised the mRNA vaccine and it actually shows very good results. So now that I've given you some basics of how people design these vaccines, let's come back to the question that you asked. So let's assume in India there is a vaccine. I think it is Covaxin. So Covaxin is prepared in the old traditional way, and what that means is you basically take the virus, you inactivate the virus, and then you inject it into the humans. So that is your vaccine. And when you inject that into humans, then the virus is dead, basically, but its structure is still intact. So the immune system thinks that there is a viral attack, and so it starts producing the antibodies. And also, the immune system makes memory cells so that it stores the memory of that incoming pathogen, so that it can make antibodies in the future. So in this step, let's say if I prepared one batch of vaccine yesterday, and let's say I'm preparing another batch of vaccine next week. So when I'm taking these viral particles this week, I'm not taking just one viral particle, right? I'm taking probably like 5 milliliters or 10 milliliters of this virus or the viral culture. And like I said, viral particles are tiny. You need electron microscopy to actually visualize this. So 5 milliliters has millions of viral particles. So if those millions of viral particles include a combination of normal strain, which is the regular strain, the original strain, and also single mutant, double mutant, or quadruple mutant, whatever, if that viral culture that I'm taking in the beginning, if it has a combination of all the strains, and I inactivate it and I give it as a vaccine, so whoever is getting that vaccine, they're protected against all the strains because the immune system has already sampled all these strains, because the initial viral culture that I've used has all the strains. And so let's say I'm preparing another batch next week, and I cannot guarantee that I will have the same composition of the virus. It could be a different composition. It may have more of the natural strain or less of the mutant strains. So if my vaccine preparation has more of the natural strain and less of the mutant strain, and when you get that vaccine, your immune system primarily prepares the antibodies and memory B cells against the natural strain, because the vaccine has majority of the natural strain. So it really depends on how they're preparing this vaccine. Are they taking only the natural strain? Then most likely the vaccine is going to fail against the mutant strains, because the immune system is not trained. But if they're making this vaccine with a composition of natural strain, mutant strains, and from North India, South India, East India, West India, then that's perfect, because you're giving the immune system a chance to sample all kinds of mutants, including the original strain. So your immune system is much more intelligent, so it makes all the antibodies that you need, and so you're protected against all the strains. I mean, the existing strains.
Okay, sir. Actually, sir, we are running short of time, and I have two very important questions from you. And in this session, I would like to first ask you that corona vaccine has been already launched in the country. Many people have been already vaccinated also, but we find that the corona cases are not decreasing despite these vaccines being given to the people. And the other thing is that the people who are actually vaccinated are showing signs and symptoms of corona, COVID-19. What is the reason of that? Is the corona vaccine working, or it's not working? What, in your opinion, it is?
So usually when we get a vaccine, we do go through the symptoms, but not as severe as the actual disease. So for example, let's take two patients. Patient number one is a corona patient. So he has COVID-19, so he has a list of symptoms. And patient number two, or healthy volunteer, let's say the second human being that we've chosen is a healthy volunteer, and he's not exposed to coronavirus, but he got the vaccine. So the way how things work is, in the healthy volunteer, the immune system still treats the vaccine or the killed virus that is present in the vaccine as an actual virus, because the immune system doesn't know if the virus is alive or dead, right? The immune system doesn't know if it is heat inactivated or killed virus, or if it is natural virus. So what happens is, you do go through some symptoms, especially the body pains, fever. Some people may have chills, cold, I don't know if people had experienced any cough, but I wouldn't be surprised if people have cough. So it is actually quite natural to get these symptoms because your immune system thinks that there is a real viral attack on the body. But another thing that I don't know if this is relevant to this discussion, but if the vaccine is not properly manufactured, like for example, let's say when you're heat inactivating the virus, if you did not follow the procedures and if you did not follow the GLP, the good laboratory practices, then there might be some viruses that are not completely inactivated. So there might be some live viruses present in the vaccine, which means you are actually infecting the human being instead of fooling the immune system. So I'm not saying that the manufacturers of vaccine are including live viruses. I'm pretty sure all these companies, the biotech companies, the pharma industry, they're following the standard procedures, and I'm sure the government of India is doing its best to support the GLP, the good laboratory practices. But the answer to your question is it's normal to see the symptoms when you get the vaccine. But then if the symptoms are persistent, and if you actually go from a list of 3 to 4 symptoms to a list of 15 symptoms, then that's a problem. Yes, so there is something wrong with the injection that you've taken. Um,
So
scientific.
you still recommend that the vaccine should be taken,
Oh, absolutely yes. Absolutely
and
yes.
it's working, and it's working.
Absolutely
Okay,
yes.
a very quick answer I would like to have for there are two vaccines in India. One is the
Yeah.
Covaxin, and another one is the Covishield, and both of them are manufactured by two different companies. Is it proper to have one dose of Covaxin and another dose of Covishield? Can it be combined?
So, as a scientist, I don't see a problem with it. But as citizens of India, we have to follow the ICMR guidelines because no matter what vaccination you are getting, you're basically trying to activate the immune system just to make sure that you're training the immune system and make sure that your immune system is ready if there is a real infection that happens. So from a science perspective, I don't think there is any difference between the two. But then, based on the I would say there are several factors because some people may have diabetes, some people may have high blood pressure, some people may have other complications such as hypothyroidism or hyperthyroidism. So when people have kidney problems, liver problems, and some people might be alcoholic, they drink a lot of alcohol, so they have liver issues. So keeping all these in mind, it's always the best thing to follow what the ICMR guidelines are put forward because they have done all the clinical studies and they're actually coming from a medical perspective. I'm just a scientist, so I can actually tell you what science tells me, but they're actually telling us with all these guidelines with their medical experience, with the real clinical experience. So that
As you said that the vaccines, production of vaccines have to be followed, certain production guidelines have to be followed, and the system of producing a vaccine is different, maybe different for one and maybe different for the other one. Does that also make a difference if one vaccine of one dose and the other vaccine of the second dose, for the second dose?
So it all depends on the initial viral culture that you're starting with, because it really depends on what kind of viruses do you have in the culture that you're taking. And when I say culture, it's basically a liquid medium in which you have the virus growing. Like I explained earlier, if you have a culture of the natural strain, then your immune system is trained only to handle the natural strain. But if your culture has a combination of all the mutant strains, then you're better off. You're in a good shape because you're training the immune system in multiple different things, so you're better protected. But then there has been some studies in, I believe, in the United Kingdom, where they found, when you're using multiple antigens, as in multiple proteins, then you may generate the immune response towards one strain compared to the other strain. So I mean, you will generate antibodies definitely, but one strain may have more antibodies generated against it, while the other strains may not have so many antibodies. But as long as your immune system is preparing or making antibodies, and as long as it is making the memory B cells that can generate antibodies in the future, we should be in a good shape.
Okay, sir. One very quick reaction. Finally, at the end of the show, that what, in your opinion, is whether the people should go in fearlessly for vaccination? There are a lot of confusions in mind. People are thinking that after taking vaccination, the person is having some thrombosis kind of thing, some blood clotting, and they have some sudden cardiac arrest due to which the person is losing their life. What is, in your opinion, are these the reason of the vaccine, or there is some other reason behind it, and vaccine is totally safe to be taken? Because this is the confusion which is in the minds of the people, and that is why we wanted to have this program, so that it is clarified. If you could clarify these quickly, so that.
Yes. So, as a scientist, I always recommend taking the vaccine. I understand what you're saying because I've read it myself in the newspapers and I've seen it in the TVs and the news. That people are confused. They're not really sure if they want to take the vaccine or if they should not take the vaccine. I would say if you have any fear in taking a vaccine, the first question that you should ask yourself is, are you healthy or do you have any complications such as diabetes? Are you overweight? Or do you have any thyroid problems, or do you have high blood pressure, or are you under stress? Because these are the factors that directly affect your immune system. And if you think you're perfectly healthy and if you don't have any of these complications, then you don't have anything to worry. Just go get the vaccine, no problem. But if you think you have any of these issues, I would recommend consulting a doctor first. Go to a doctor, a clinic, and discuss your medical situation. If you have a stressful life, talk to your doctor and see if you are okay to get the vaccine, because stress is one thing that would create several complications. And in fact, stress has been shown to influence the immune system. So if you're stressful, definitely that will slow down your immune system. And when you're going through a pandemic, you cannot take chances if your immune system is going slow. It has to be at its maximum speed, which means you need to reduce the stress. And I'm not going to recommend doing yoga or meditation. Okay, so those are not the only solutions for this. You need to identify what is it that makes you that calms you down. So for some people, just watching TV will relax them, or for some people, reading books will relax them, or for some people, going for a walk or going and sitting in the beach for 5 minutes will relax them. So you need to identify what is it that's really relaxing you, and do that so that you can calm down and reduce your stress. So I would say, please go get the vaccine if you're healthy, and if you have any secondary complications, please consult a doctor and take advice, and then go for the vaccine. But my suggestion is please get vaccinated.
Okay, sir, this is the last reaction I want from you, last comment on this. Uh, is this the vaccine which we are now taking, is totally safe and does not have any side effect or after effect, maybe after 10 years or 15 years or 20 years? Do we expect any kind of after effect with these vaccines?
Usually, I don't think so. I don't think there will be any side effects or complications. But my statement completely depends on how good the vaccine is. And when I say how good the vaccine is, what I mean is, the company or the pharma company who is making this vaccine, did they follow the GLP? If they followed the GLP, then there should be no problem, because the COVID-19 vaccine is not the first vaccine that human beings are taking. We've taken hundreds of vaccines, and in fact, most of us are vaccinated when we're infants, less than 5 years old. But we're doing perfectly fine. We never have any complications. For example, polio vaccine or BCG, and there's several different vaccines, and we've got injected with those, and we're doing just fine, and we are completely healthy. This is the same thing for coronavirus too, because it's all dependent on your immune system. How healthy are you? How healthy is your immune system? That is what makes a big difference, whether the vaccine is going to be effective or whether it's going to work properly or whether it's not going to work properly. Because if your immune system is not getting trained well with the vaccine, then there's no point of taking a vaccine. So that's why I suggest, if most of us have stressful lives, especially these days, going to the office in the morning on a two-wheeler through this carbon monoxide clouds on the traffic, that itself is a lot of stress, right? So this is something that we cannot do anything about. The environment, is completely polluted. So there are so many stresses that we're going through. The body is going through. Just calm down. And if you think you're healthy, you have nothing to worry about, go get the vaccine. If you think you have some, complications such as blood pressure or diabetes, I would highly recommend. The first thing you should do is get a healthy lifestyle, consult a doctor, get advice, and then there should not be any side effects or long-term effects of these vaccines, as long as you're healthy and your immune system is healthy.
Okay, thank you very much, Dr. Ravikiran, for joining us this evening and making people aware of the facts of the coronavirus vaccine and the coronavirus itself. We'll be soon back with you with some more questions on viruses and their effects on human beings. Till then, we thank you very much once again for joining this program. You have been watching the DN News, the DN Talk Show, a special program that we talk with eminent personalities in the country. We'll be right back with some more issues, with some more talks at the same channel, same time, again on Sunday. Till then, see you. Goodbye. Thank you.